Okay, so I'm going to talk to you now about, we're going to just keep growing our thing. So we're going to go from looking at aligning two sequences. Now we're going to align, a whole bunch of sequences at the same time, and talk about the problems that we get with doing that and stuff like that. Actually, I think for this one I'm going to use a laser pointer. Let's see if we can use the laser pointer. Maybe a little bit easier to use. Okay, so first I'm going to talk about the multiple sequence alignment. Then I think we'll have time. We'll talk about different things we can do with a multiple alignment, different types of things we can do, looking at motifs, profile patterns, hidden Markov models, and so forth. Okay, so first of all, let me just kind of tell you generally about the multiple alignment thing. This is a little bit more history and so forth of what people do. Like I said, first Needham and Wunsch started doing this stuff like in the 1970s. They were doing single alignments, and like I said, that's where Smith and Waterman came up with their procedure for doing local alignment. And then in the 1980s, you started to see more and more methods develop for multiple alignments. In particular, there's methods that, for instance, a lot of tree building approaches and so forth. And of course, nowadays you see even more of these things coming around. And now the problem with multiple alignments is that you could imagine trying to optimally align three sequences very much like I described. For two sequences, you would sort of get the best alignment of the three up to a point, and you would keep adding on. Now, of course, as opposed to that sum matrix we looked at, we would have a sum cube, right? We'd have a cube, and then obviously if we try to do four sequences, we have a hypercube and so forth. The problem with that, of course, is that it doesn't, as we'll talk about when we talk about how these problems scale, it doesn't scale very well computationally. It's way too expensive computationally to do that. And we'll talk a little bit about that issue when we talk about fast alignment and computational complexity. But that problem becomes untractable. So people came up with more heuristic approaches that we're going to talk about now. And the goal of the heuristic approach is kind of what's shown here, to basically take 30 sequences, align them up, find particular positions that are highly conserved and so forth. And actually, people found this notion of the positions that were conserved and variable so important and interesting that there's actually software developed that just doesn't even show you the letters in the columns. They just showed you the coloration to describe the conservation patterns. So it turns out that those are very important in terms of thinking about the key residue of protein, the active site, or how a protein might fold and so forth. So the basic heuristic that people came up with to solve this is pretty simple approach. It's just a kind of progressive approach. It's a clustering type of approach. We'll talk about the different ways they approach, but the simple idea is you would align, say, two sequences together well. You would lock their alignment after you did that, and then you would add things on to that and so forth. A little bit sort of similar like this solving the subproblems type of approach, but now it's for looking at multiple sequences. But one of the issues here is that, and this sort of different from the dynamic programming thing, when you solve the subproblem and then added things on, you don't have any guarantees that when you add something to the alignment, the optimum of that thing added on really wouldn't disturb the solution to the subproblem, as we'll talk about. And so that's why these are much more heuristic, and you don't know if they get an optimum solution. Um, so the different approaches people had. One of the most popular programs a while ago was called Cluster. It uses this method called UPGMA for unweighted pair group method with arithmetic mean. It's a verbose term, but basically a lot of these procedures, they had to make decisions that were kind of summarized by here. You know, when when they were joining sequences together, say they had a group of sequences, they had found two and they added one on, they would now come to the next sequence. And so they could do different things. They could say, oh, is the next sequence in a cluster already? Okay. Or in a separate cluster, should they glom those two clusters together? Well, they could look at just the linkage of one sequence in the cluster to another cluster, or they could look at the average of all the sequences in one cluster to another cluster, or they could demand that every sequence in one cluster would match up with another sequence above a certain threshold to get merged. And these are different approaches, you know, complete linkage, simple linkage, whatever. And there's a number of problems here. One I've kind of alluded to already, and that's really the main problem is that as you're aligning things together, in all these approaches, you fuse the alignment of two things together to create like a consensus sequence from the two. And then you add things onto that thing, and then you fuse them as you add them. But what if when you've, say, created an alignment of maybe 7 sequences, you come to the 8th sequence you want to align onto there, and that changes how you might want to align within those 7. You can't do that. You've already frozen that alignment based on the earlier ones. And so you can see this kind of greedy way this is kind of done. It creates a potential problem. And people often use this term as a local minimum problem. So you're kind of finding the immediate minimum, but that might not be the global or best minimum for the whole alignment. Another issue is that, remember, we were talking about the substitution matrix and how it's different for globally, that sequences are very different from very far. If you imagine you're trying to align together sequences from a very diverged group, you know, they might have some that are very close to each other, some that are very diverged. You know, you don't know really that ahead of time that well. So often you have to use the same set of parameters, like the same substitution matrix in this thing. And so this kind of creates this global parameter choice. And you can imagine a way of getting around that. But in general, usually the multiple alignment subset starts off with a single set of parameters for the whole bucket of sequences, even if within that bucket, there might be some that are really somewhat closer than farther together. And then there's another problem too, and this is very unique to protein sequences, and that's what's called the domain problem. And that's, imagine you have this sequence here, and you start with this, and you match it to another sequence. Okay, so now you get an alignment, but they only align over a portion of the sequence. That's the bit over here. And now you get a third sequence. Oh, and it aligns, say you do simple linkage, and it aligns to that second sequence. And so you lock that on too, but maybe it's not aligning to the same part of that second sequence that the first one is. It's kind of like this type of thing. So now, you know, you've obviously created a mess here. And this is, you can sort of see the problem. Now, if you come to a fourth sequence that actually has parts of the first sequence and the third sequence conjoined, it's going to have a very hard time making a good match here, right? Because it's not going to know what to do here. It's going to get a bit messed up. And this is called the domain problem. Okay, so that's a kind of quick go through of the notion of how we might align multiple sequences. And now what I'm going to do is talk about what do we do with multiple alignments. And that's actually one of the most important ideas, I think, in computational biology is when we have a collection of data, we can kind of fuse that collection of data to create some pattern. Because one of the key aspects of biology, as you all know, is that there's a lot of data, but there's a lot of redundancy in that data. And a lot of times what people are really doing in biology is looking for kind of a summation of a group of data. So we talked about this in the first sort of lecture. We talked about the number of protein sequences and genome sequences going up, but the number of underlying shapes or folds isn't nearly going up as much. It's kind of plateauing. And the way people get at this is they create these this notion of a kind of pattern and an abstracted pattern from kind of growing collections of data. And this is quite useful. And it's also very useful from an efficiency point of view, because if you imagine you're aligning and putting together more and more things, it's extremely computationally expensive to look at all the individuals within that group. It's much more efficient to look at some abstracted pattern. So there's a variety of methods people have for building patterns, and it's useful, again, to talk about. These are classic methods. So the methods I'm going to talk about are motifs, profiles, and hidden Markov models. And the idea basically is you're given a sequence alignment like this, and you want to summarize that sequence alignment in some abstracted language, okay? That you could, understand the sequences better, or it could be useful from an efficiency perspective in storing it into a database. So first, let's talk about the notion of a motif. That's the simplest type of thing. And I should point out, I'm talking about this in the framework of we're given we're given a group of sequences of, say, a protein. Another very common application, which is a little bit more genomic, which I'm not going to go over, but uses the exact same method, is often there's groups of sequence in the genome, for instance, like transcription factor binding sites, that share a common pattern, okay? And we want to, again, get the motif for the pattern. In fact, that's the somewhat more common case nowadays than the looking for a motif in a protein. But I'm only going to cover the protein one, but it's almost exactly the same as the DNA case one. So the motif concept is pretty simple. We might have here's an alignment. I'm going to be showing you the same example alignment over and over again. And, we might look at the alignment and we might see, looks like, let's see, do you see what you can see here? So yeah, this column here is completely conserved. It has an F, whatever we say, we would say it's conserved. The motif is obviously an F here. But then we would also find, for instance, that some other columns aren't perfectly conserved, but almost conserved. So this column is mostly an F, or it's a Y, and F and Y are very similar amino acids. So maybe that would make sense. It's some sort of generalized aromatic amino acid. And here, for instance, we have an I, a V, and L. So again, we have the notion of just a hydrophobic amino acid there at some point. So we'd create some notion of conserved columns and then conserved kind of patterns we have. And then people make databases of this. One of the early versions of these databases called ProCite. And it would have these little patterns of, conserved sequence motifs, which actually are biologically very useful. So they would give you a hint as to what this group of sequences is doing. And if we were looking, for instance, an analogous case for DNA sequences for, say, a binding site, they would give you a sense of what's the core part of the site. And then you could imagine coming up with the syntax for this. This particular database used what's called a regular expression type of thing. This is where you would describe the motif with a regular expression. The key aspect of this is you could write the pattern down in some easy to understand language, and that communicates to people and so forth. And that will be in contrast to what we'll be describing in a second. So now let's talk about the notion of a profile. So a profile is a more abstracted computer notion of a pattern. And here what we do is, and this was originally developed, I think in the probably the 1990s or so by this guy, David Eisenberg, who's, again, a big figure in the early development of this field. He had the notion of if you have an alignment, counting up how many times you have each of the different 20 amino acids. So for instance, in this column, A occurs once, R occurs a lot of times, whatever. In this column, blah, 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 you have 4 Cs, and so forth. And you would just add them up, and you could sort of see these as these are the frequencies or probabilities of this particular letter to occur. And so you'd end up with this matrix now, this profile matrix that would be the length of your aligned columns versus 20 for the 20 different amino acids. And obviously, this profile subsumes most of the motif thing. You could kind of get the most common thing here by just taking the max. You could, sort of develop some other notion of consensus. Another thing that you could easily do is you could look at how variable a column is. If you have a column that has just one number, it would obviously be not very variable, whereas if it's split equally over all the things. And people often use this idea of sequence entropy, and we're going to be talking about just how you calculate this this type of quantity a little bit in a little bit. And here's an actual profile for this particular thing. You can see your sequence here, and you have your different amino acids, and you can, for instance, see you have a conserved cysteine here. You can see it's how it would be represented. Here's now if you wanted to convert this this profile, you often want to convert it maybe into a kind of a log odds or kind of probability type of thing, where again, what you'd want to do is you'd want to compare the number of times you found an amino acid in a given column versus some form of expectation, right? If you have obviously alanine, it's much more likely that you'd have it there. So you want to somehow downweight that particular expectation. So you'd have some weighting of that type of thing. And then also, if you calculate the frequencies that things occur in columns, as I said before, it's a very easy step to compute what's called the Shannon entropy of that column. And that's basically the sum of the frequency, or P ln P, which is the Shannon thing. So the kind of surprisal that you would get from any given column in your, or any given, the emission of any particular letter is the logarithm of its probability, and you'd weight that by how often you'd see that to get the overall entropy, or the overall surprisal that you have in that particular column. So now, if you had some simple profile, which was maybe normalized, right, you could imagine using this in a little bit more of a rigorous way to scan for related sequences. You could imagine taking this type of profile and going through an unknown sequence. And now, of course, you wouldn't have to match it anymore to all the different sequences in your database, which had built your profile. You would simply say, oh, the first thing is a C. What's the score I get for the C in the profile? The second thing is a G. What you would just read off those scores you would get from matching that substring to the profile. You would write that down. Then you could graph that overall score that you'd have as you slid this profile over the sequence as like a window. You would take the profile and slide it over the sequence, and then you would compare the overall score you got for matching it in any given positioning the window at any given point, and you would compare it to a threshold, and you would basically say you have a match if it's above a certain threshold, or you don't have a match if it's below a certain threshold. There's a number of programs that use this kind of profile building approach. One of the most widely used ones is this program called SciBlast, which was a a generalization of the BLAST program that we're going to talk about subsequently. And it would basically build a profile up of your sequence matches and then use that profile to search the database and grow the sequence matches. And I'll just say a little bit about that because it talks a little bit about some of the issues that come with building these up. And let's see how we're doing for time. Okay, so the way this particular program would work is you'd have a database of sequences, for instance, and you'd have a query sequence, and you would compare your query sequence to each sequence in the database, okay, using just say Smith-Waterman alignment and so forth. You could do it slightly differently, but to keep it simple, you could do it with Smith-Waterman, and you would find if there's a match. And sometimes there would be a match, and sometimes there wouldn't be a match, and so on and so forth. And then you would take all the sequences that have a match, and you would find the matching region. You would overlap the matching region, and you would calculate in this overlapped region a profile, from that. And so that would become your this profile. Now this becomes the query. So now you could take this thing and now query it back against all the sequences, okay, the way we just talked about. And the idea here is now since you've generalized this this thing a little bit, it will maybe match to more things. So now in addition to matching the things that went into the profile, maybe this thing will match these additional sequences now shown in yellow. Okay, so now you do the game again. You take these new sequences plus the other sequences, you align them up, you make a little profile of these things, and you have a new profile, this yellow profile, and you keep iterating this process over and over again. Now, the advantage of this is a number of things. First of all, potentially, because the profile generalizes beyond the individual query sequence, you're going to find more things than your query would. And the other kind of advantage is that you get this pattern now. This in addition to the query, you get this nice pattern that the query is part of. So that's a kind of nice method. Now, one issue that comes up is that sometimes you get convergence. Convergence would mean that you would take this profile, you would find matches, you wouldn't find any more matches. You would just find the same matches that went into a profile, and then you would say the matching has converged, and then you'd be done, and that's good. But you could also find an explosion where you keep iterating this procedure, and each time with the new profile, you drag in more and more things forever until everything in the database is matched. And obviously that's nonsensical. And that would be a kind of situation where you had polluted your profile potentially. And one aspect of profile pollution that you have to think about is if you think about nucleotide sequences or protein sequences, often there's a kind of an entropy you might compute for them. They have a kind of complexity to them, and they kind of look complicated. But there's also in the database, there's just runs of A's, runs of L's, just long sequences that people describe as homopolymers, low complexity regions, and so forth. And you can use this F-lon F characteristic to find them by looking at the composition of bias within a window and sliding that window to 5 low complexity regions. And these regions obviously are a big headache in database matching, and they kind of confound all the sequence matching that I've talked about, which is really predicated on your sequence has a kind of structure. It's as if you're matching normal English text and you're expecting a sort of complexity in the language where you have John likes fish and Mary likes this. You're not expecting A all the time, and that's going to screw up your whole approach. So what people often do is mask these sequences out or treat them in a somewhat different way. So now what I'm going to talk about is I've kind of talked about the notion of I give you an alignment, I build a profile from that alignment by counting the number of things in each column. but the alignment process is separate, right? That's from the UPGMA clustering, and then they build the profile. But we could imagine a process where the aligning and profile building process are happening at the same time. So, and this is a little bit like the sort of local optimum issue that we were talking about earlier, where we were refining the profile and at the same time refining the alignment and doing that kind of together. And so people have developed a number of approaches for doing this. They fall under the umbrella of expectation maximization and Gibbs sampling. And so I'm going to talk to you about these two approaches now briefly. So basically, the expectation maximization, again, this is a very common statistical technique that's used in many, many contexts, much more generalized from the specific thing that we're going to talk about, but it's very good to get a good grip of how it works in this specific problem so that when you come to another problem in someplace else in bioinformatics or in just data science in general, you can come back and use your intuitions here. So expectation maximization basically has two steps. There's called the E step, where you figure out the expectation of given a likelihood, including the imagining that there's some latent variables, you're sort of fixing them at a particular thing. And then a maximization step where you re-estimate these latent variables. You find their maximum likelihood estimates for these parameters or latent variables. Okay, and so you kind of, and what happens in EM is you kind of iterate between these two different type of things. And so, again, I think if you describe it, that's the general idea, but it's good to, if you describe it in a very concrete context, you'll get a lot of intuition for this. So basically what you're going to do is you're going to generate kind of a profile matrix, and then you're going to you're going to match it against the sequences. That's basically the alignment problem. You're going to align sequences to it, find things to add into it. And then you're going to re-estimate your profile matrix from the things you just pulled in, kind of like a little bit like the SciBlast type of thing going on and so forth. And so that's the simple idea. You're going to re-spin that around and around. That's EM. Now, Gibbs sampling is a kind of slightly more, I would say, sophisticated way of thinking about this problem than EM, and I'm going to describe that in a little bit of detail here. So in Gibbs sampling, what we're going to do is when we come to the finding of the sequences and the re-estimating, we're not necessarily going to pick the best match all the time or the most optimal match. We're going to pick that most optimal one with some frequency, but we'll also allow some less optimal ones to come in. And in that way, we're going to get a somewhat more generalized profile. And I'll walk you through how we do this. And like so, I'll just walk you through the whole procedure of doing this for a bunch of sequences. So you imagine, first of all, we're going to have a bunch of sequences, and we want to iteratively estimate the profile or the PWM or the matrix from those sequences and find the profile instances, the motif, the pattern in all the sequences. So we want to align them up. So we might start off, we'll just randomly build our pattern. Okay, just start random. Okay. And then what we'll do is we'll remove one sequence from it. We'll try to define the weight matrix from that thing. And we can think of the weight matrix as finding the probability of each sequence. And now we're going to pick, for the sequence that we've left off, we're going to align the profile to that sequence. But this is the key point here, and that is the one highlighted here. We're not going to necessarily pick the best match. We're going to look at the probability of that sequence aligning against the profile say positioned at any given spot, and we're going to pick in accord to that probability. And the way to think about it is the reason it's used Gibbs sampling. We're imagine we're kind of picking according to like the sort of Boltzmann factor. We're picking according to a probability distribution. And so here's so for instance, in this context here, if we have this, if we take these matrices, we leave off one sequence. We take these sequences, we leave off one, we make the matrix, we estimate the parameters of the matrix. Now we can slide it against that sequence. And here's how likely, or how the score of it matching at any given point. So it's most likely going to match here, but it also could match here with some score. So what we're going to do is we're going to then pick the place it's going to align in accord to the relative weights here. So we'll pick this spot, say 50% of the time, and this spot, say 20% of the time, in accord to its thing. Then once we get that match, we'll re-estimate the weight matrix, and then leave out another sequence and continue this process. I think that these procedures have somewhat different advantages or disadvantages. You can see Gibbs sampling is a little bit of a generalization of EM, where you're not necessarily picking the best match. One of the issues is it doesn't really converge the same way, right? Because you can always pick a low probability type of thing. So that's a disadvantage. I mean, the advantage of Gibbs sampling is it provides a sort of more robust optimum, more global optimum, because you're not always just picking the best match or something. You're potentially picking off the best match. So it potentially provides a better solution. And so now I'm going to talk to you. I think I can just fit in, I have 3 more minutes. I just want to fit in the hidden Markov models very quickly. So hidden Markov models are a further generalization of profiles and EM. Here, what happens is you imagine having your sort of profile column, and you imagine it kind of emitting amino acid sequences in accord to a probability distribution. But now you have the idea of going from one of the columns to the other to be associated with a kind of a probability of transition between the 2 different states. And so this allows you to handle the gaps, insertions, and deletions in sequence very probabilistically and nicely. And what people often think of is they think of the underlying symbols that you observe, and then they think of the states, that those are essentially the columns that generated those states. And so they think of these as the state sequence is kind of like a hidden choice, and then the observed symbol is what you observe. And you're trying to fit often the columns that these states to the observed sequence. And so the nice thing about this formalism, we'll talk about a little bit more, is it's a very general formalism, and you can really incorporate the transitions between many different match states and so forth. And I'm not going to go into this in some detail, but one of the key things is a little bit more complicated formalism, but it uses a lot of the same ideas. So for instance, the best path, the most probable path through your model, it's solved by something called the Viterbi algorithm, which is a somewhat more complicated type of thing than we've talked about. But to some degree, it's actually very much similar to that simple dynamic programming thing that we talked about. There's another type of algorithm that's very common here called the forward algorithm, which it finds not the best path, but the summation of all the paths to the model, which is something we haven't talked as much about.